So let's understand the penicillin production process flow that happens typically in a pharmaceutical industry. This is a general block diagram for any drug manufacturing. A typical generalized or a generic process flow where we have number of reactors. A reactor is nothing but a properly controlled chamber. Okay, a chemical reactor we normally call it, because we add some reagents to it so that some reaction takes place inside under a given controlled temperature, pressure, level, and other parameters. Okay, so in a typical process, you may have reactor 1, reactor 2, and reactor 3. Actually, when you add reagents to a reactor under controlled condition, the reactor can be a fermenter. The reactor can be typically it is a reactor can be a fermenter in pharmaceutical industry, or it can be any chemical process equipment. Okay, or in unit operation that we have generally seen. So the usual process is likely like this. No, you have a reactor where you are adding the group of reagents under a controlled conditions through a medium, and then the extract of this is given to a filter. It can be a manual filter, or it can be a chemical filter, or a biological filter, or a vacuum filter. So there are different types of filters. Okay, again you are adding solvents to the filter. The output of this is given to a dryer. Dryer is again a unit operation where it is a hot air chamber. Okay, where you are allowing the waste gases to go out, and the solid particles you are giving it to another reactor 2, where further you are adding more reagents here. Further chemical reaction takes place. The output of this again given to another filter. Again you are adding solvents, and this output is going to another evaporator, and through the waste is going, and the output of this is given to the distillation column. You are adding solvents here, waste is going up. Then the output of this distillation column 1 is given to another reactor 3, where you will see in every reactor you are adding the reagents. Okay, so that the further chemical reaction takes place. And again you have it is going to the distillation column 2, and where we are adding the solvents again, and waste is going up. It is coming to filter 3, the next filter. And the filter, again the output of this is going to the crystallizer 1, where we are adding more solvents, and the output of the crystallizer is given to centrifuge. And we have seen already what happens in a distillation column, what happens in crystallizer, what happens in centrifuge, and the control of centrifuges, control of crystallizers, control of distillation columns. All of these we have already seen. So here the chemicals are actually coming through different phases, and then they are going to another filter 4. Then again it is going to the dryer. The output of this is coming to the wet granulator. We have seen what is a typical granulator. Granulator is nothing but it dissolves instantly when you make it a granulator, because it is coming after the dryer. It can be a spray dryer, or it can be a hot air dryer. So under controlled conditions, the entire product becomes a granulator. Granulator is nothing but what we see, the granules normally in capsules, okay, or the instant coffee. All these will be in granule forms, so that they can dissolve instantly. And the capsule, if you see, the top cover capsule is basically made of protein, so that the inside granules of drugs gets dissolved immediately along with the protein. Then again it goes to dryer 3, and again you have the blender, which is nothing but a mixing of all these things. Then you are pressing the tablets for different dimensions, then tablet coater. Then we are getting the tablets, because tablet coating is very important again here, so that it doesn't immediately when we put it on the tongue, it should not it should not dissolve immediately in the tongue itself. So that's the reason why we have a coating for the tablet, and similarly we have a capsule for the granules. Okay, then we have so this is a very general block diagram of making any particular truck. So if we go next deep into the thing, the production of antibiotics, it happens mainly the fermenter. We will see what exactly is a fermenter. The output of the fermenter is taken through preliminary treatments, and then it is filtered, then liquid comes. Okay, after filtration, the solids go to the extraction, continuous extraction part. The extract part of it comes back to join the liquid, and the waste solids of extraction go to a dryer, and this dryer, the waste solids after drying, moisturizing, they're all sent to animal feed supplements because they go on to be added as a supplements for animal feed. So the extract which is coming from the continuous extractor is joined again after the solvent extraction part, and which is a partially purified antibiotic in solution. Then you have a recrystallization purification, then we are getting the antibiotic. So this is a very, very general production of antibiotics. So let's understand the penicillin from its accidental, we call it accidental discovery because, it was actually, we will see it in the next thing, where a Scottish biologist in 1928, Alexander Fleming, discovered that Staphylococcus culture he had mistakenly left growing in open was contaminated with a mold which had destroyed the bacteria. Okay, so then he thought, which mold is getting the bacteria destroyed, and why it cannot be acted as a antibacterial medicine. So otherwise we normally clear the whole thing, but this that's the reason why penicillin is always considered to be, it is a discovery by accident. Okay, so he was leaving a particular mold at one particular place, and which was killing the bacteria. And the mold is nothing but what we call Telugu as buzu. Okay, that kind of a thing. So after isolating a sample and testing it, he found that it belonged to the penicillin family. Later, the mold was classified as Penicillium notoneum. At first, it was difficult to convince people about its potential uses, but gradually, people realized, because that was the discovery which was used massively during World War II, that led to Nobel Prize for Medicine in 1945 by rightful owners. Penicillin has made its proof as a miracle drug that revolutionized the course of the medical industry. Okay, without further ado, let's take a look at an important aspect that made penicillin accessible at large, its industrial production. So if you understand what is penicillin, it is a naturally occurring antibiotic ever discovered. Okay, you don't require it. It never came through a chemical reaction. No, it was a naturally occurring antibiotic. It's a group of antibiotics produced by Penicillium fungi. It is a group of closely related compounds, not a single compound. That's the reason why we have amoxicillin, ampicillin, phenoxymethylopenicillin, and so there are around 50 drugs that are basically of penicillin's family. So this is exactly what was there. And once he did it in 1928, but later in 39, 11 years later, powdered form. Okay. In 1945, they successfully treated a human. When it is tested on a human, we normally call it as in vivo. If it is tested in a glass tube, we normally call it as a in vitro in chemical terminology, in biological terminology. In vitro means it's a test tube sample. In vivo means it's a human sample. So that is the reason why many of the COVID samples COVID vaccines are still in vitro stage, in vivo stage, right from in vitro stage too. And final testing always has to be on animals or human beings. So that led, in 1943, they produced penicillin on a large scale, which helped many to treat many casualties during World War II with bacterial infections. Okay, so if you briefly see the raw material for this penicillin production is corn steep liquor. Corn is corn steep liquor is nothing but a solution with concentrated corn in it. Then you have water for nutrients. and calcium carbonate for pH adjusted, because this corn strip liquor always has to be adjusted with nutrients and pH. Okay, the nutrient salts you further, whatever we do not have in water, like zinc sulfate and sodium nitrate, we also use them. And then oxygen is the major thing that is reacting with, to give the, it reacts with some carbon compounds and gives away the carbon dioxide. Then we have mold spores, which are normally inoculated. Inoculated means it's basically the vaccine kind of a thing. Okay, under controlled conditions. Then we will see in the process how phenyl acetic acid, amyl acetate, chloroform, and disodium phosphate and sodium acid phosphate, how these are all used in a process. We will see it here. And just a media formulation, if you see, pH should be around 6.5, temperature between 20 to 24 degrees centigrade. You should have oxygen in the medium. Nitrogen, which is a corn strip liquor, you should have 8.5% nitrogen in the corn strip liquor. Glucose should be 1% in the media, 80% ethanol, then phenyl acetic acid, propane acid, lactose 1%, calcium carbonate 1%, sodium hydroxide, sodium hydrogen phosphate 0.4%, and anti-foaming agent like vegetable oil. So all these are the media that is basically used. And just try to follow this process flow chart for the production of, if we describe the process of penicillin production systematically, you will have a fermentation medium here on top. A suitable fermentation medium, such as corn strip liquor, adjusted for nutrients and pH, is inoculated with selected mold spores, which multiply to produce penicillin. That's the basic chemical formula. Inoculated means it's like a vaccination, injected with a limited amount. That's what we normally call it as inoculated. So when suitable fermentation medium such as corn strip liquor is when it is adjusted to nutrients and pH and inoculated with selected mold spores, actually you have the penicillin production. So the culture is charged into carbon steel fermenters with an operating capacity of 5,000 to 30,000 gallons. Okay, so these fermenters, vertical pressure vessels, because pressure vessels means they are maintained under controlled pressure and temperatures, have a height to diameter ratio of 2 to 1. If the 2 meters vertical and 1 meter diameter, or 3 meters vertical, 1 meters diameter. It has an operating pressure of 5 to 15 psi gauge. The fermentation medium is sterilized by heating it under 15 psi steam for 30 minutes, following which the tank is water cooled continuously. We will see the actual diagram of the fermenter in the next slide. When the medium has reached a temperature of around 24 degrees centigrade, it is aseptically inoculated with mold spores from top again. Okay, the mold having already been prepared in vitro means in glass, by breeding a selected strain of P. cricogenum on agar slots and seed tanks. So that's exactly what is happening. To initiate fermentation, the medium is agitated and sterile air is blown through the tank sparger. Frothing is minimized by adding an anti-foaming agent such as octadecanol, and temperature of 24 degrees centigrade is maintained constant by the circulation of cooling water through the coils around that. The aerobic mold is allowed to grow for 5 to 6 days. It is aerobic because it's in the presence of oxygen. Anaerobic means in the absence of oxygen. So the aerobic mold is allowed to grow for 5 to 6 days, during which time the carbon dioxide is evolved. Okay, because these are all the carbon fermenters. So the carbon dioxide is evolved after reaction with the oxygen that is present there, and then you have the air and carbon dioxide escaping from bottom. So this is exactly the major reaction that happens in fermenters. So then onwards, it's a process of extraction. When the optimum amount of penicillin has formed in the fermenter, the fermented liquor, what we normally call liquor, is nothing but a concentrated solution with a given solute containing large quantities of mycelia, large quantities of mycelia, which is nothing but the vegetable portion of a fungus, is called mycelia. It is and relatively small amounts of penicillin is passed through a stainless steel heat exchanger, which we normally and then through a vacuum filter, a rotary vacuum filter, where the mycelia are removed and discarded from the bottom. The clear filtrate known as beer may contain as much as 4,500 international units of penicillin per millimeter. Whatever we are getting it from the continuous filter, it is then given to continuous extractor. Here the concentration of penicillin is considered to be 4,500 international units of penicillin per milliliter of the solution. So here international units is nearly 1 microgram is equal to 40 international units. That's how we have to remember. 1 microgram of is equal to, normally the fat soluble, you have vitamins, water soluble and fat soluble. And for water soluble, no problem. But for fat soluble, like vitamin A, D, and E, and other, some hormones, you have to give in micrograms. You cannot take it in milligrams and grams. So when they are given in micrograms, they are also measured in terms of international units, where 1 microgram equals to 40 international units. Okay. Likewise, you can calculate the total composition. So when we get the clear bead of penicillin here, this may contain a 4,500 international units of penicillin per milliliter of the solution. Then the penicillin is removed from the crude liquor by a continuous extraction process. The pH of the crude beer is adjusted to about 2.5 with dilute 10% phosphoric acid, which is sent from the top. The liquor is then passed into continuous extractors, where the penicillin is extracted by amyl acetate. When you add amyl acetate, the penicillin is extracted and sent to the neutralizing tank. The reaffinate containing the water-soluble impurities and some of the solvent is sent to solvent recovery column, where it is further processed. And the amyl acetate is removed here, and the waste goes to the bottom. The penicillin containing extract is then channeled to a neutralizing tank, where a buffer solution of sodium phosphate and sodium acid phosphate, disodium phosphate and sodium acid phosphate are added. Penicillin passes into buffer solution, which is separated from the amyl acetate by supercentrifuging at a speed of 15,000 RPM here, and the solvent is recovered and reused here with the removal of amyl acetate again. Okay, and this again goes to a continuous extractor. The aqueous buffer solution of penicillin is acidified here and conveyed to a second series of continuous extractor, where it passes counter current to the another solvent, which is chloroform here, or it can also be a amyl acetate. That's a solvent again. We are adding the solvent and again further processing it. The extract from the whatever is the extract after solvent recovery form. So this extract is coming to another solution tank. So the extract from the second extraction is mixed with a solution of sodium hydroxide or potassium hydroxide. So the sodium salt of penicillin readily dissolves in water. The separation of the immiscible liquids is accomplished by supercentrifuging again, and the concentrated and purified water solution is blown through the biological filter to remove bacteria. The resulting sterile solution is dried in a freeze dryer in bulk or in containers by freeze, vacuum spray, or electronic drying. And after this, you are getting a bulk penicillin. So this is the total process of penicillin. You will see in the very beginning block diagram that we have seen, it is a combination of different chemical reactors and adding you are adding the solvents at different stage, extracting it, solvent recovery system where you are recovering the solvent, because you are adding the solvent amyl acetate here, recovering the solvent, adding the chloroform here, recovering the chloroform here. So at different stages and the under controlled conditions, you are finally getting the penicillin here. So this is exactly what happens in a most important chemical reactor. In this case, is a fermenter. You will see you have a paddle wheel here, which are nothing but stirrers, and you have the steam inlet from top and food inlet, whatever is coming in here. You have a penicillium inlet here coming into the. So you have a cold water jacket all around the fermenter. This is a cross-sectional view, and you have a temperature recorder and cold water inlet coming in, and then you have the cold water outlet after getting circulated. Okay, so air, it is purged into the thing for because you are stirring it through purging of the air and through the stirrers. You have a motor for stirring. You have a pH controller reservoir on top, steam outlet, and then exhaust outlet. And this is a paddle wheel. And finally, this is the harvest. Wherever you take penicillin or anything that you are extracting, this is the harvest part. If you see the another configuration of this will be a overall what you will have a stirrer. You have a cooling water out, cooling water in. You have a supply water, supply air supplying here, which is giving the thing. Penicillium and other nutrients in a medium, you are giving it here. Air and carbon dioxide is escaping through this. Cooling water is coming in, cooling water is going out here. Steam for sterilization, if you want, you are supplying the steam here. And then penicillin is taken out. So this is what exactly happens in a fermenter. And there are some important aspects of this penicillin production is you have to maintain a very good transfer of oxygen across the liquid interface. That's very important because that's where the oxygen, because it's an aerobic digestion, aerobic fermentation. Heat transfer metabolism as a process tends to give off heat achieved through cooling jacket, whereby cool water is passed through in order to maintain the temperature. Bulk flow and mixing is achieved by impellers, bubble columns, batch and continuous culture. How nutrients and substrate will be delivered to a culture in a reactor is normally what we call it as a batch. Steam is used to keep the reactor running aseptically, because that's where a temperature of 121 degrees centigrade and a pressure of 15 psi for 15 to 30 minutes sterilizes the whole culture initially, and then it is maintained at 24 degrees centigrade for fermentation for 5 to 6 days. If you see the media formulation, microorganisms require carbon, hydrogen, oxygen, sulfur, and nitrogen for cell growth and cell maintenance, and also require small amounts of nutrients, trace elements such as copper, manganese, and cobalt. Frequently depend on these are all given by water source. Okay. So that's what is there. And if you see the production process of a typical antibiotic, streptomycin, and again, this is a different process. You have a master culture going through the agar slopes. Okay, you have a shaker flask, getting to seed vessel, and then it is the fermenter. Okay, here we are getting the basic process, and then it is going to acidification, filtration, neutralization. Acidification can be a continuous extraction, because you are adding a solution and extracting through it, and extracting that amyl acetate kind of a solution. It is going through the filtration, neutralization tank, clarification, ion exchange reactant, then evaporator or dryer. Then it is going through the crystallization, vacuum oven for dryers. Then calcium chloride crude complex is given. Again, catalytic for crystallization. Then once it comes out, it is going through the freeze drying, and there you are filling it to the vial. Injection vial, we normally call as a vial. It's a glass tube where the injection is there. Then it is capped, sealed, labeling, packing, and dispatch. This is how a streptomycin injection is made. Okay, so that's all about the penicillin production. What we have seen it in this diagram, and I think now the penicillin production process is clear, where the instrumentation plays a very important role in maintaining the pressures and temperatures at different reactors. Okay, and this is all about the penicillin production.